Good afternoon, everyone, and welcome to today's webinar entitled Uncovering the Neural Circuit Mechanisms of Spontaneity. I'm David Page, a faculty member here at Whitehead Institute. In a few moments, we'll hear from our presenter, Whitehead Institute Valhalla Fellow Allison Hamilos. First, I would like to let you know that this webinar is being recorded for future viewing. Also, we will be taking and answering questions at the end of today's presentation, but you can submit a question at any time during the seminar using the Q&A box at the bottom of your screen. So with that, I'd like to introduce our speaker today, Allison Hamilos. Allison earned MD and PhD degrees from Harvard Medical School as a member of the HST, Health Sciences and Technology program. She completed her PhD dissertation with John Assad studying nigrostriatal dopamine circuits in mice going about their daily business. She also completed an HHMI Med Fellowship with Jeff Karp, designing surgical devices for transplanting stem cell technologies for the treatment of type 1 diabetes. Allison holds bachelor's degrees in chemistry and biology from MIT, where she did undergraduate research with Professor Kathy Drennan in X-ray crystallography and with Professor Monty Krieger in cholesterol metabolism. Allison is a systems neuroscientist and Whitehead Institute Valhalla Fellow. Her lab asked the question, and it's what we'll hear about today. Her lab asked the question, how do we choose when we can't know the right answer, or if the right answer might not even exist? The philosopher Al-Ghazali reasoned free will lies in our ability to act under uncertainty. Free will in situations where we can only pick randomly at best. Intriguingly, such stochastic behavior may be the key to intelligence. Machine learning algorithms depend on stochasticity, and probabilistic cognitive models explain human reasoning in situations where artificial intelligence struggles. So what neural mechanisms allow us to behave spontaneously? Allison's lab aims to answer these questions by dissecting the role of neural circuits disrupted in neurological and psychiatric disease. Drawing on clinical insight and tuition, Allison's lab trains mice to carry out complex behaviors. She trains mice to carry out complex behaviors. She does neurosurgeries to record and manipulate neural circuits with optogenetics, and she uses computational modeling to understand how these circuits give rise to spontaneous behavior. In this webinar, Allison will discuss how her lab is using clues from human disease to understand how our brains enable spontaneous behavior. She will demonstrate how the moment-by-moment signaling of dopamine neurons influences both our perception of time and our ability to self-generate movements. In addition, she will explain how these insights can help us better understand the many mysterious symptoms that arise in neurodegenerative conditions such as Parkinson's disease. So Allison, welcome and thank you. Take it away.
Thanks so much, David. All right, share my screen. I think you guys can see. Yes, we're good to go. Awesome. All right, everyone. So for the next 30 minutes, I'd like us to start by thinking about an old mystery and somewhat philosophical question, which is what makes us move? What goes off in our brains that allows us to decide when to spur the body into motion? Well, here's what we know. We know that when we move, thousands of neurons in higher motor centers suddenly begin firing together in concert, and this strong, coordinated neural activity gets relayed via spinal cord motor neurons to muscle fibers that power movement. But I was told this in med school, right? But this never was satisfying to me because of course, it does not explain why those neurons started firing together in the first place, which I think is kind of the crux of the issue. Now, one idea is that this activity in our brains could come from sensory areas. For example, if I throw this football to you, that strong activity charging through your visual pathways could get passed along to the motor centers that trigger the coordinated activity that's needed to move. But not all movements result from these kinds of overt sensory cues. So, for example, consider a cup of coffee that might be sitting in front of you during this talk. I know I have mine. So, presumably, it's been sitting there long enough, it's no longer causing bursts of sensory activity in your cortex. But mysteriously, at some moment, your hand may reach out, pick it up, and take a drink. Now, here it's not so simple to imagine where this motor activity comes from. I will operationally refer to these two classes of movements as reactions and self-initiated movements, whereas self-initiated movement is distinguished as one in which the brain has to rely on some internal process rather than some abrupt sensory cue in order to generate it. Well, it turns out it's not just our intuition that tells us there's something mysteriously different about these two types of movements. In fact, clues from human movement disorders also suggest these movements are generated in a different way. In particular, you might be familiar with Parkinson's disease, which is a movement disorder in which patients feel slowed down and also have difficulty initiating movements. But what might surprise you is that patients particularly struggle to self-initiate movements, like picking up this cup. But if a strong sensory trigger like a football comes flying at them, they can actually move in reaction to it as quickly as a healthy subject. Now, this mysterious phenomenon is called paradoxical kinesia, and it's so striking that assistive technologies have been developed to actually exploit it to help patients. So, for example, there are walkers that display visual cues on the ground that tell patients when they should be taking steps, and patients can react to them and actually walk better. Now, the striking difference in the way in which these two types of movements are affected in Parkinson's suggests whatever is going wrong in this disease might just be a critical clue to figuring out just how the brain spontaneously initiates its own movements, seemingly like of its own volition. So, what might be going on here? Well, we've known for decades that there's a profound loss of dopamine neurons in Parkinson's disease. Now, these neurons live in a small strip of your brainstem called the substantia nigra. You can actually see them by eye from their dark pigment here. So this healthy midbrain at the left has lots of dark dopamine neurons, but this midbrain here at the right, which has Parkinson's disease, has completely lost this dark strip, showing us that these neurons have almost completely died off. Now, the fact that these neurons are missing in Parkinson's suggests they must normally be vital to this process of self-initiating movement. But precisely how this degeneration gives rise to the symptoms of Parkinson's remains poorly understood. And it's actually been surprisingly difficult to ascribe a role for healthy dopamine neurons to the movement initiation process. In fact, back in, around 2016 or 2017, it was shown that when dopamine neurons are stimulated to fire at physiological rates, this fails to elicit movements. And that suggests that whatever dopamine neurons are doing in generating self-initiated movement must be something more subtle than directly triggering it. But there's actually one other important observation from Parkinson's that might give us a clue as to what's going on, and that's this mysterious slowing down. So not only do patients move slowly, but even their perception of the passage of time is slower than reality, as if their internal clock were somehow ticking too slowly. This phenomenon can actually be measured with a variety of timing tasks, and here's one you can try yourself at home. So grab a stopwatch and grab a friend. And when you're ready, tell your friend you're stopping the watch by saying start, and then have the friend say stop when they think 5 seconds has gone by. Now, if your friend is a perfect timer, they'll say stop right at 5 seconds, just like the picture. But as you might guess, no one's timing is actually perfect. What will actually happen is your friend will say stop a little before or even a little after 5 seconds, but they won't be exactly right. Now, keep track of when your friend moved this time, and then try repeating the task again. You'll find on the second trial, your friend will actually move at a slightly different time than the first. But nevertheless, if you repeat this game lots of times, you'll find that despite the natural variability in the response times, your friend actually responds pretty close to 5 seconds on average. And this variability, this is normal timing behavior for healthy subjects. But now, what if your friend has Parkinson's? Well, in this case, you'll notice something very strange. If you repeat the procedure, you'll find that your friend will move measurably later than expected, both on individual trials as well as on average, as if their internal clock were somehow running slower than your stopwatch. Now, intriguingly, this kind of distortion of internal timing, it's not just seen in Parkinson's. In fact, you could give a healthy friend a drug that reduces the ability of dopamine to signal in the brain. And in that case, you'll also cause these late shifts in behavioral timing. Now, these dopamine blocking drugs, they might sound kind of esoteric, but actually they're really commonly used in both the clinic and hospital settings for many neurological and psychiatric conditions, including bipolar disorder, schizophrenia, Huntington's disease, Tourette syndrome, even delirium. These drugs can relieve symptoms in these diseases, but fascinatingly, they can also cause side effects in these patients that look just like the symptoms that we see in Parkinson's disease, such as causing difficulty moving and slowing down time perception, like we see here. But the plot actually thickens because it's not just knocking down dopamine that affects movements and timing. In fact, several neurological and psychiatric disorders are treated with drugs that increase dopamine signaling, like levodopa in Parkinson's disease, or stimulants like those used in ADHD. Now, fascinatingly, if your friend takes levodopa before doing the task, you'll actually get the opposite effect to what you see in Parkinson's disease. So, drugs that increase the amount of dopamine available in the brain will cause your healthy friend to move too early on average, as if their internal clock were sped up. So, for nearly a decade now, I've been just fascinated with these mysterious movement and timing symptoms that we see in Parkinson's disease, as well as other neurological and psychiatric conditions, as well as in the setting of drugs that affect dopamine signaling, how they can also cause these strange effects in healthy people. So, how does this all happen in disease? And what can we learn about these symptoms, about how healthy neural circuits in the brain work to produce spontaneous movements and decisions more generally? Which, I'm using the word spontaneity kind of loosely here, right? That can mean a lot of things to different people. To me, I'm saying spontaneity to describe something that to me feels like it's an internally generated process, as opposed to a reaction to something in the environment. And I think of these types of movements or decisions as fascinating because they really seem like the sorts of decisions and things that we do that make us who we are, right? They come of our seemingly of our own volition. So they're existentially fascinating. But beyond philosophical interest, and perhaps more importantly, if we could somehow understand how this all works in the brain by linking dopamine's mysterious effects on spontaneous behavior in both health and disease, we could potentially develop better ways to treat and prevent neurological and psychiatric diseases as well, both of which we still just really don't understand. Now, these mysteries have served as a launchpad for the broader work in my lab here at Whitehead Institute, where we study dopamine neurons and the neural circuit mechanisms that underlie these kinds of spontaneous, internally driven decisions. So today I'm going to share with you the first steps towards unraveling these mysteries, which actually go back to when I was an MD PhD student at Harvard working with John Assad. I'll show you how we found that the normal signaling of healthy dopamine neurons can probabilistically influence when our spontaneous movements happen. And that might sound a little bit abstract or technical right now, but by the end of the talk, you'll have a really good intuition for how this works. I'll then build up to the current work that my lab is pursuing, which is built on this foundation, where we're using these insights from pharmacology and disease to understand how we produce spontaneous motor, cognitive, and perceptual decisions more generally, in both the setting of health as well as how this might go wrong in the setting of disease. Okay, so before I get into the new stuff, I want to first set the stage with some past work that I did as a PhD student, trying to figure out how dopamine's roles in movement initiation and timing might be mechanistically linked. Now, because others had already shown that dopamine alone was not sufficient to trigger movements, we suspected that rather than acting like a motor neuron, which can cause muscles to contract, maybe what dopamine neurons were doing was something more subtle. Maybe dopamine neurons instead guide when spontaneous movements occur by controlling the timing of when an intended movement actually unleashes. So, to test this hypothesis, we recorded and manipulated dopamine neurons in awake, behaving mice trained to make spontaneous self initiated movements. But to study self initiated movement in a meaningful way, we had to create a situation where we could feel more confident that we were actually looking at a spontaneous movement as opposed to some reaction to the environment. That is to say, we needed a scenario where the mouse had to rely on some kind of internal process rather than an abrupt sensory cue to decide exactly when they would move. To satisfy this constraint, we trained mice to do a novel self-time movement task, which is basically a timing game for the mice, just like what I showed you earlier that you could play at home with your friends, where mice received an audiovisual start cue and then could earn a juice reward if they correctly timed their next movement to happen during a hidden time window that happened several seconds after the cue. Now, the difference between the mouse task and the human task is instead of saying stop, the mice would instead indicate their guess of timing by licking a juice spout. And just like in the human game that I showed you earlier, the mouse would only get one attempt to report their timing guess per each exposure to the cue, meaning if they licked too early or too late, they would not get any juice rewards on that trial, and it would in fact have to wait like 10 seconds before the next trial before they could get to try again. Okay, so one thing it's really important to understand here, right? There is a cue, but the point is that the animal is not being cued exactly when to lick. Instead, she has to estimate internally elapsed time to decide when to make her move for several seconds. By that time, whatever sensory activity is generated by that cue has died down in the cortex. So this is some internally driven process. Now, because she has to rely on this own internal process of self-timing, these movements implicitly fall in this umbrella of what we are operationally defining here as self-initiated or internally driven. Now, one other thing to note is because timing is implicitly built into this task, it's actually exactly what we need to test this hypothesis that dopamine neurons might control the timing of self-initiated movements. Now, like mice, so like humans, mice are pretty good at timing, but their time behavior is even more variable than ours. So for instance, the mode of this mouse's timing distribution is about 2.5 seconds. But notice that sometimes he happened to move a second earlier, and other times he happened to move whole seconds later. This is while the mouse is trying to time the same interval. So this is natural variability that happens. Now, it's important to note this variability is expected, but it still might make the mice seem kind of sloppy at timing. But for the purposes of our experiment, this variability is not a bug. In fact, it's a vital feature to the task because it's exactly what we need to test our hypothesis. With it, we can ask, what are dopamine neurons doing differently on these trials where for some reason the animal decided to move early versus these other trials where he decided to move late? And specifically, based on our hypothesis, do dopamine neurons have higher signaling activity and release more dopamine on trials where the animal moved early compared to these late trials? To answer this question, we took a fiber photometry approach to record dopamine neurons in the substantia nigra, which remember, that's the population of dopamine neurons that dies off in Parkinson's disease. Now, if you're not familiar with fiber photometry, don't worry, you don't have to understand all the details here to understand the talk. The basic idea is that it allows us to record fluorescent signals that are related to the activity of specific kinds of neurons, in this case, dopamine neurons. Now, when I was, starting out grad school, I always thought the term activity was a little confusing. So if you're not a neuroscientist, maybe this is a little weird. By neural activity, neuroscientists mean how much are these neurons firing electrical signals called action potentials that are part of the process that neurons use to communicate with each other. So this is a bit of an oversimplification of this, but basically the way you can think about this is the higher this green signal is over here, the more the dopamine neurons are potentially signaling to downstream neurons. That's the way we think about this. And remember, they do this by releasing the neurotransmitter dopamine. That's why they're dopamine neurons. To measure these fluorescent signals, we use a viral genetic technique to express this green fluorescent calcium indicator called GCAMP6F in dopamine neurons using a DAC-Cre mouse, transgenic mouse, and this Cre-dependent virus. And then we surgically implant a fiber optic cannula in the brain, which you can think of as like a light telescope that lets us look deep down into the brain and collect light from these neurons where they live in the substantia nigra. Now, if all of this was confusing to you, just remember, the brighter the green signal that we measure through these fiber optics, the more active the dopamine neurons are thought to be, the more they might be potentially signaling to other neurons. And that's all you really need to know to understand this talk. Now, for any experts out there, just a heads up, even though we're measuring GCAMP signals here, which this is a calcium indicator, I'm going to refer to all these photometry signals as dopamine signals just to make things easier to follow for a general audience. So, if I'm being a little bit loose with my language, that's why. Now, also, just know as well, if you're an expert, that we co-expressed a second calcium-insensitive fluorophore called TD tomato, and that allows us to correct for any movement-related artifacts that might be happening in our optical signal. And if you're not an expert, and if those last two things I said didn't make sense, don't worry about it. You don't need to know. Okay, now I want to first show you the simplest possible thing that we see when we record from dopamine neurons in the timing task. This is the average of the fluorescent GCAMP signal across all trials for one mouse, where the first lick time was between 0.7 to 3.3 seconds. But the plot's a little complicated, so I'm going to break it down. The x-axis here is time, but there's actually two parts to the plot. The left is a cue-triggered average where all the signals have been aligned to the time of the cue and averaged together. And this is to show the responses of dopamine neurons that are related to the cue. But I also want to show responses related to the movement. The problem is that the movements happen over a really broad range of times relative to the cue. So we can't just plot everything aligned to the cue. Everything would get very blurry. So what we did here is we split the x-axis so that I could plot the signals here aligned to the time of the movement on the right side of the plot. Now, the signals here are unfolding over the course of seconds. Just keep that in mind. Let's now focus on what we noticed right away. We saw that there was a cue-related increase in the dopamine signal, and we also saw a movement-related increase in the dopamine signal. And neither of these signals was too surprising, because dopamine neurons were already known to fire in response to salient cues and at the time of movement onset. But what was surprising was the third thing that we saw, how the signal appeared to ramp up from the time of the cue up until the time of the movement onset, starting several seconds before the mouse actually moved. Now, the rising of the ramp made us think of a timer, so we asked whether this ramp might be related to the mouse's natural variability in its timing. To test this idea, we pooled the dopamine signals by the time of the movement relative to the cue. So here we're looking at trials where the first lick happened between 4 to 4.25 seconds after the cue, and we notice the same rough patterns that we saw before. Now, it gets interesting when we add in another movement time. So this purple trace shows all the trials where the first lick happened between 1 to 1.25 seconds. Now you can see a few differences. First, baseline levels of the dopamine signal start from a similar level, but you'll notice they're a little bit different even before the cue happens. The position at the cue onset is higher for the earlier trials. The ramp doesn't dip down as low on the early trials as it does on the late trials. And the early trial ramp is steeper than that for the later trials. But despite all of these differences, in both cases, the dopamine signals ramp up to a similar level immediately before the movement onset. Now, it's important to remember that the differences between these two traces here, these two sets of pooled trials, are the result of the animal's own natural variability in its timing. We didn't do anything to change the task. Now, if we add in the pooled trials that happened in between, we see that we can predict by eye when the movement's going to occur, starting more than 4 seconds before the movement actually unleashed. Now, that's a long time for neurons. So just think about this for a second. Just by looking at what dopamine neurons are doing, you can learn something about what the mouse is going to do seconds into the future, which I think is kind of crazy. Now, we saw this pattern in every mouse that we tested. When we average together signals across all 12 of our mice, we find that this pattern becomes abundantly clear. And I'll just note here for any experts out there that although I'm showing you GCAMP signals, which are calcium signals related to the firing of dopamine neurons, we also measured striatal dopamine during the task with fluorescent dopamine indicators, including D-light and DA2M. And with these, we observed similar ramping photometry signals using these methods. So we can say that these ramping GCAMP signals actually do reflect downstream dopamine release. Okay, so because dopamine neurons are communicating different signals to downstream neurons in trials with different movement times, we asked whether these signals could predict the timing of movement initiation in a quantitative way. So to do so, we focused our attention on dopamine signals that occurred before the movement onset. Now here to more cleanly show the pre-movement responses in this plot, I'm showing you the same data from the last plot, but I've truncated the traces from our 12 mice just before the movement onset. Now, by eye, you can see a couple of predictive features in the signal. We see the signaling of dopamine neurons is predictive of the movement timing even before the cue happens, both via this baseline offset, where the blue traces here that correspond to the earlier movement times are systematically higher in terms of the dopamine signal even before the cue. We also see this predictive relationship during the timing interval via these ramping signals that we saw before. Now, for any experts out there, our TD tomato control data didn't show either of these patterns, and that was really reassuring because it showed us that the baseline offsets and ramps that we see in the photometry signals are not explained away by optical artifacts. Now, because the dopamine signal looked reminiscent of a timer, we asked whether a simple threshold crossing model for these signals could predict when the movement would happen. Now, the way that this model works is that we imagine that dopamine neurons undergo a sort of reset at the time of the cue, after which their signaling builds up towards some threshold level, which when crossed triggers the animal to move. And of course, on average, you can tell by eye this model works great. We found that the time at which these dopamine signals crossed a variety of arbitrary thresholds strongly predicted the movement time, both on average here as well as on single individual trials. So although I'm just showing this model for one threshold, you can see by eye that we could have drawn our threshold anywhere along the y-axis with a similar result. So we conclude that dopamine signaling does in fact quantitatively predict when self-time movements will initiate via this apparent ramp to threshold process. And just an aside, again, for any experts out there, I'll just note that that our generalized linear models showed that these signals were not explained away by ongoing body movements or optical artifacts, which suggested that these ramps and baseline differences could in fact be related to a more abstract internal process related to self-timing. So given all this, we asked whether the signaling of dopamine neurons could causally influence movement timing. To do so, we optogenetically activated or inhibited dopamine neurons on 30% of randomly interleaved trials. To specifically activate dopamine neurons, we used a similar viral genetic technique to what I showed you before in the photometry experiments. But here, instead of expressing a fluorescent protein, we instead expressed an activating opsin protein. Now, if you're not familiar with optogenetics, all you really need to know is that opsins are light-gated ion channels, kind of like those that are found in your retina. So when we shine light onto these activating proteins, it causes them to increase positive charge inside the cell, which depolarizes the neuron and activates them. So this technique lets us increase the activity of large populations of dopamine neurons at specific moments during the task. To do so, we pulse blue light onto these dopamine neurons from the time of the cue all the way up until the time of the lick, which increased dopaminergic activity while the light was turned on. We confirm this by also co-recording dopamine release with fibrophotometry. In a separate cohort, we also specifically inhibited dopamine neurons by expressing an inhibitory opsin protein called GTACR2, which basically just does the opposite thing. Now, given that our dopamine signals look like a ramp to threshold, we hypothesized that activation might cause more early movements on stimulated trials, whereas inhibition might cause more later movements. So to test this idea, we compared the distributions of movement times on these stimulated trials and compared those to the interleaved unstimulated trials. Here I'm showing these movement distributions as cumulative distribution functions, or CDFs, which you can just think of as the integral of the movement time histograms or distributions that I showed you earlier. The blue curves here are stimulated trials, that's the blue curve, and the black curves here, these are the interleaved unstimulated trials. But I always think this is a little hard to see on Zoom, so I shaded the area between the curves here so that it's a little bit clearer, and I also drew this arrow just to be able to show you the sign of the effect. Okay, so consistent with our expectations, optogenetically manipulating the firing of dopamine neurons bidirectionally tune the timing distribution. And here's how we know this. Activation caused early shifting, which we see by the fact that the stimulated CDF here, it rises faster than the unstimulated CDF. And we also know that this means an early shift because the stimulated CDF lies to the left in our CDF plot of the unstimulated trials. So overall, this means that the animals made more early movements on trials that were stimulated compared to the unstimulated trials. Now, by contrast, inhibition of dopamine neurons caused the opposite effect. It caused rightward shifting in the stimulated distribution to later movement times, which looks kind of like what we see in Parkinson's disease, what I showed you at the beginning with the human game. But unlike activation or inhibition, no opsin control animals showed no consistent effect of stimulation. And this is a really important control for us because it shows us that the changes in timing that we see when we activate or inhibit are not some trivial result of the animals just reacting to the stimulation light. It suggests that there's actually something different about the dopamine system that's impacting the behavior. So together, this shows us that dopamine neurons can causally and bidirectionally influence movement timing. And really excitingly, this seems a lot like the picture I showed you at the beginning, where the drugs that increased or decreased dopamine could push the movement timing distribution around. But this experiment showed us two additional things that we didn't know before. First, that healthy dopamine neurons can push the timing distribution around too. And this can help explain the natural variability that we see in timing. But second, and more mysteriously, there was something much more interesting going on here than initially meets the eye, which I'm going to explain. Okay, so although these manipulations could tune the movement timing distribution, when we tried to activate dopamine neurons in the same mice, in the same way, outside of the context of the task, this didn't make them lick. So here the mice were just sitting on the platform, just hanging out, and we'd zap the neurons here during this green window, but we see that the mice did not start licking. Now, likewise, inhibiting dopamine neurons outside the context of the task also did not prevent the mice from licking. So we actually were giving these animals these sort of randomly timed, surprise juice rewards just to get them to lick at baseline, but their licking didn't change when we inhibited during the red window here. So that's kind of weird. In summary, whether we activated or inhibited dopamine neurons, we just could not seem to influence lick initiation outside the context of timing. And this was the case both for naive mice that had never done the task, as well as expertly trained mice that we'd been training for several weeks. This was consistent with what others had seen, which is to say that dopamine was not acting like a motor neuron here. It wasn't causing muscle fibers to contract. Rather, it suggested that dopamine neurons were doing something more subtle and more interesting, I think. It seemed instead that there was something very special about the moments in which the animal was in the process of self-timing, as if the only way we could get dopamine neurons to influence movement initiation was if the mouse was already in the process of deciding when it was going to unleash this planned movement. But the effect of stimulation was not to cause the mouse to initiate movement at one specific time, but rather to tune a set point of what was an apparently probabilistic movement distribution. Now, for this reason, we wondered if rather than specifying a precise movement time, maybe dopamine neurons were instead modulating when movement would occur by tuning a probability that a planned movement would unleash at any given moment. Sort of like how rolling a set of dice in certain board games can probabilistically decide whether you get to move on a given turn. I thought this idea was really interesting. And because our curiosity was piqued, we went back to our photometry data set and asked whether the dopamine signals that we measured in the context of normal behavior could also predict the probability of movement initiation at any instant in the timing task. So to do so, we derived a probabilistic movement state decoding model, which probably sounds like a bit of a mouthful, but it's actually pretty simple. So I'm going to break it down. The way it works is that on any given trial, as long as the mouse has not licked yet, she can be in one of two states, either not licking or licking. At each moment in time after the cue, she can either stay in the no-lick state, or she can decide to transition to the lick state, thereby initiating movement. So what does this look like in our data set? Well, if we take any individual trial and examine it using this framework, it means that we can assign a lick state to every point in time between the cue and the first lick, which we can plot down here in our state plot as a 0 for each not lick state and a 1 for each lick state. So to build the model, we assigned a lick state to each point in time between the cue and the first lick. And we assume the mouse only enters the lick state once on a given trial, and that this happens right before we detect the movement. We did this for every time point and every trial in our dataset. We then decoded the probability of being in the lick state for every movement time t, given that the mouse had not licked yet. So here, just for example, I'm showing this time t as a red dashed line, but keep in mind that we calculated this probability for every time point in the dataset. Now, to determine whether dopamine signals could encode this conditional probability, we then derived a nested logistic lick state decoding model, which is even more of a mouthful. But here's the idea. Okay, so this included the dopamine signal at each moment in time t. And the idea is that you want to predict whether the mouse is in the movement state from this dopamine signal. But we also wanted to incorporate the richness of the ramping and baseline offsets that we observed. To do so, we included the history of the dopamine signal in these 200 millisecond windows of time relative to time t, which we represent as just the average of the dopamine signal during those points of time. So all these go in as predictors to this big regression model. But just keep in mind, the goal of this model is to see if these dopamine signals can predict the probability of licking, given that the mouse has not licked yet. Now you might be thinking, okay, hold on. Probability of licking, that all seems really abstract. Even if the model works well, what's the ground truth for a probability of licking in our data set? Well, it turns out that the instantaneous probability of licking, given that the mouse hasn't licked yet, is just by definition a quantity called the hazard function, which is something that we can measure really easily from the mouse's behavioral distribution using a statistical technique from survival analysis. So the predicted hazard function, which I'm showing here, h of t, can actually be decoded from our logistic regression as the average across all trials, where there are a big n number of trials per session, of the probability of being in the lick state at time little t on trial little n, given that the mouse was in the not lick state at time t minus 1, and given the level of the dopamine signal as well as its history at time t. So our job here is to use the results of our logistic regression to calculate a predicted hazard function and then compare this to the true hazard function for the session. So here I'm showing the mean true hazard function for our 12 mice calculated across 98 sessions, where each thin line represents the average for a single mouse, and the thick black line is the average across all sessions for all mice. Using our model, we find that the moment-by-moment dopamine signals were actually highly predictive of each session's hazard function and explained 65% of the variance on average. Now, it's really important to note that the probabilistic model that we built here, it has no idea what the true hazard function actually looks like. This is not a regression fit to the hazard function. Rather, we're predicting the hazard function using the dopamine signals alone. So how likely is this model to work by chance? Well, as a control, we derived the same model again using the same data sets, except this time we shuffled the time points during the trial. And what this did is this abolished the predictive power of the dopamine signal in predicting the movement state, which we see reflected in the fact that the decoded hazard function really has no systematic structure and only explains about 5% of the variance on average. Now, the red and black curves here, the point is, if this all sounds like just a lot of words, the point is the red and black curves here, they don't match anymore, right? So although altogether here, these results show us that dopamine signaling during this task strongly predicts the moment-to-moment probability of self-time movement initiation, even in the context of normal, healthy behavior. So when we take this together, the photometry, optogenetics, and modeling results, we can conclude that dynamic dopaminergic activity modulates the moment-to-moment probability of self-time movements. So what does this all mean in the context of Parkinson's? There is an interpretation here, okay? So it seems like what's going on is that dopamine signaling is gradually increasing the probability of movement initiation over the course of the timed interval. So we suspect that Parkinson's patients live in a state of low movement probability, much like the mouse when it moves relatively late, but much more extreme, in which the baseline dopaminergic drive is just so low that even when patients make a big effort, the system just can't ramp up very easily, thereby delaying movement initiation and sometimes causing movement initiation to fail altogether. Now, we might take this a step further and suggest that folks taking drugs that increase dopamine, like amphetamines, might also be more like the mouse when it moves relatively early, but again, more extreme, with such high dopamine drive at baseline that the system revs up too easily. So putting this all together, this might suggest that dopamine neurons typically operate in this intermediate regime in healthy subjects, but that pathology can drive this dopamine ramp to threshold process into these extremes that mess with our propensity to move. This suggests a new unifying explanation for the motor and timing deficits in Parkinson's disease. Now, this is a great takeoff point for us to start talking about future directions. And to do so, I'd like to start by pointing out a few remaining mysteries. So first, what makes dopamine's influence probabilistic? And second, why should it be? Because in principle, this seems to prevent us from timing precisely. So isn't this some kind of bug? What might probabilistic signals like this be good for in the brain? So during the last few minutes together, I'd like us to take a stab at these questions. And in the process, I'd like to introduce the vision for the research program that my lab is working towards here at Whitehead Institute. So what might probabilistic signals be good for in the brain? Well, from a computational perspective, random signals are incredibly useful to the field of computational cognitive science. In particular, Bayesian probabilistic models of cognition have made machines that can make intuitive decisions under uncertainty in ways that feel deeply human, like this machine, which can predict from this ambiguous image whether this tower of blocks is stable or if it's going to fall. And the way that it does this is by imagining or literally probabilistically simulating what might happen next by using draws from a probabilistic generative model. And by doing so, the machine can make intuitive judgments about the tower that match human judgments surprisingly well. Probabilistic signals also form the guts of some of the biggest advances that we've made in artificial intelligence. For example, deep neural networks completely rely on probabilistic signals to explore and tune network weights when learning to solve problems in high-dimensional spaces. Now, this is all computer stuff, but even from the intuitive perspective of your own experience, the truth is, we know that we don't want to do the same thing all the time. In fact, sometimes the ability to make random choices can be the difference between life or death. If you're this mouse trying to evade this owl, if your movements in the next few moments are predictable, you're dead meat. Instead, the mouse's best chance is to choose unexpected movements and unleash them at unpredictable times, with which she'll hopefully buy herself a reprieve. Now, this is an example of how a probabilistic signal might be useful in the movement domain, but random signals can also help us in other hard situations, like when we're forced to make difficult decisions under great uncertainty. Now, here's a classic thought problem that illustrates this, which was proposed by the 10th century Islamic philosopher Al-Ghazali, who came up with this example while trying to understand free will and where it comes from. He asks us to imagine that you're starving, and on the brink of death, you're offered a choice between two completely identical pieces of fruit. Now, if you needed some reason, like some external cue or physical quality of these pieces of fruit to decide which one to take, well, you'd just sit around all day deliberating and you'd die from starvation. But Ghazali points out that even in this extreme case, when we have no information to go on, something inside us will still let us pick one. And intuition tells us we just pick randomly. It's not even hard. Now, while this might seem like a really extreme example, the truth is we're faced with choices every single day where we can't know the right answer, or the right answer might not even exist, where intuition tells us our decisions may very well come down to a coin toss. But the point is, we don't need a coin to do this, right? We can make random choices all on our own, and we can use these to spontaneously come to our own decisions. Now, so far, I've shown you examples of motor and more abstract choice decisions, but even in the sensory domain, we make probabilistic perceptual decisions all the time. Like one night, a couple years ago, I was driving in the snowstorm on 95 South, and I swear I had to basically imagine where the lane markers were. I had to basically rely on my expectations of where they might be and my best guess as to the likelihood of the scene if the lines were in any one spot. Like, usually lane markers don't go like this, right? So there are these three types, at least, of probabilistic decisions that I can think of off the top of my head. What movements to make and when, which option to choose, and whether or not you perceive something in the environment, which all have in common the need to act decisively under uncertainty. And they're all decisions where we have to act spontaneously in real time. So the question that I have is, could there be some connection between the probabilistic signals that I observed in the dopamine system during my PhD and these types of probabilistic signals that we make, or probabilistic decisions that we make every single day? Well, I think so, because as a med student working on both neurology and psychiatry services, it became clear that it's not just spontaneous motor decisions that are impacted in Parkinson's. It's actually all kinds of these internally driven probabilistic decisions, like the kind that I've shown here, these abstract choices, and even perceptual decisions, which are modulated by these dopamine drugs that increase or decrease dopamine signaling in the brain. We see that these types of decisions are perturbed across all these conditions that we notice are treated with these different types of dopamine drugs. This leads us to a hypothesis that dopamine is a neurotransmitter that's capable of acting as a tuning dial for spontaneity by modulating the degree to which we behave randomly across all these domains. And I'll just say, I was pretty surprised when I realized in the clinic that this just really isn't the relationship between dopamine, or rather this relationship between dopamine and what I'm calling spontaneity. It's just not the way that people generally think about dopamine or these diseases where folks are taking dopamine modulating drugs. In fact, conditions like bipolar disorder and Parkinson's disease are considered so different, they're not even treated by the same kinds of doctors. But taking these patterns together with what I'd observed in the dopamine system and timing, I started to think, maybe what this is telling us is that there could exist some shared dopaminergic circuit mechanism underlying our ability to behave spontaneously across all these modalities, one that might unite the way that we think about these different diseases under a common mechanistic understanding, and one that might help us find better ways to prevent and treat them. I also wondered, could the probabilistic behavior of this system be the key neural circuit player underlying probabilistic models of cognition, which are making huge waves in AI? These questions are the foundation of my research program. So to start to answer them, my lab is beginning by establishing a common behavioral framework for studying spontaneity in mice across the motor, cognitive, and sensory domains. In particular, we're training mice to do behavioral tasks where they have to make decisions under uncertainty in each of these domains. We're then applying intersectional viral genetic techniques to specifically record and causally manipulate genetically defined neurons at multiple sites within decision-making circuits in these animals while they're doing the tasks, including dopamine neurons. We're then relating the activity of these neurons to the animal's trial-by-trial variability in their decision-making. At the same time, we're also collecting behavioral data from humans while they do the exact same games and tasks that the mice do, which we're combining. We're combining the results from our mouse tasks and our human tasks to better understand how what we see in mice might relate to what happens in humans. And you can actually play these games yourself by logging onto our website. It's hamilos.wi.mit.edu, and you can click on the Games tab here, and you can actually play these games for fun or to help us out with our scientific projects. So if you'd like to hear more about the mouse or human experiments that we're working on, or if you have ideas for how these concepts might synergize with your own ideas and interests, please do reach out to me afterwards. I'd love to brainstorm with you. So with that, I'd like to thank the wonderful team that was part of my PhD work, particularly my thesis advisor and longtime partner, scientific partner, John Assad, as well as the students who are currently working in my lab at Whitehead Institute, who over the last year have started pulling down some amazing and exciting new data sets from both mice and humans making decisions under uncertainty, whose work I didn't have a chance to show in much detail today, but which, we hope to be releasing some new results in the coming months with preprints on bioRxiv, and we'll also be making announcements about that on our website. So I'd like to also thank at this point the Valhalla Foundation for supporting my lab and the Whitehead Fellows program more generally. Their transformative gifts have created this tremendously unique situation for us where we get to be, what we like to think of as kind of hardcore systems neuroscientists immersed in this Whitehead community, which is just an unparalleled place for biomedical innovation, where we're learning so much from the amazing folks around us and really starting to think about behavior, not just from what's happening in the brain, but also thinking about how that connects with physiology in the body. I'd also like to thank Mary Sullivan and Monica Concepcion for putting together this special webinar, for bringing us all together, and especially David Page, who introduced me at the beginning, for being just a tireless supporter and hugely inspiring biomedical scientific mentor. We're so grateful to get to do this exciting work here at Whitehead and to get to share it with you all. And we're just looking forward so much to presenting some of this new work in the coming months. So please do stay in touch with us via email. My email's up here at the top. You can follow us on Twitter or X now, where we'll post our preprints and also update you about new games for the human tasks. And with that, I'd love to take some questions. Thank you so much.
Okay, Allison, thank you for a very stimulating and spontaneity-provoking seminar there. That was great. I just want to remind all of you who are joining us today to post your questions in the Q&A. And those questions are coming in. So let me begin with one of the first questions, Allison, which is a question about circadian rhythms and whether the pathways that you're discussing, whether they intersect with circadian rhythms and whether the circadian rhythms might play some role in conjunction with what you're describing in convincing the brain to initiate movement.
That's a great question. There's a lot of ways to answer that, and I will tell you a few things that might be thought-provoking about that. First of all, the brain has different timing systems. In fact, any neuron can, in some ways, be thought of as like a timekeeper. But the big systems that people usually talk about are the dopamine, nigrostriatal dopamine system is involved in kind of timing on the order of seconds. There's another system in the brain thought to exist in the cerebellum that does sub-second timing. And then, of course, there are these broader, longer time intervals, over the rhythms of the course of a day, like circadian rhythms, which are thought to be controlled by, different circuits, including those in like the hypothalamus. Does it intersect with what we see in these these kind of seconds long timing decisions? I would be reluctant to say that any sort of decision that we're making isn't influenced by these types of rhythms. I think that at different times of the day, right, you have such different input to all the parts of the brain from different ascending modulating systems, where, certainly I think maybe our behavior at different times of the day is going to be a little bit different in terms of our perception of time. But, I think the way I would answer that question is by saying that, yes, it is thought that circadian rhythms are controlled by a different system, but it probably also influences the way that the dopamine system works. I will add one last thing to this, which is, as a grad student, I tended to be somewhat nocturnal. Um, I cannot tell you off the top of my head that I've noticed any difference in terms of the dopamine signals or the timing behavior of the mice, depending what time of day that it was recorded from. But we also didn't check that in great detail.
Okay, so that you haven't seen that the steepness of the ramp is a function of the hour of the day, for instance.
No, but it's an interesting question. We could go back.
Right. Okay, a couple of questions coming in or taking shape in my own mind with regard to disease and Parkinson's and such you mentioned. So you mentioned that the way that your experience in the clinic is that physicians and physician scientists who are taking care of patients with Parkinson's or treating individuals with dopamine modifying drugs are not thinking in the way that you are thinking and presenting stuff to us today. So could you compare and contrast? What is that prevailing worldview? Or is it just a black box? Is there a model that in a sense you're seeking to replace? Or are you seeking to create a model where there is none?
It's a good question. I'd be reluctant to say that no one in the world thinks about things this way. But what I will say is, being a student on the wards and kind of, seeing the types of patients that are interacted with on both of these services, right? As a neurology service, we weren't really taking care of psychiatry patients. There is some intersection with some folks who have neuropsychiatrists. I don't mean to say that, nobody is thinking about things this way. But what I will say is, I think what I describe here are things that you get some intuition about working in the clinic. I think most people who've gone through medical training think, oh, isn't it interesting that we use these dopamine modulating drugs for so many different things? I think that the way that we're thinking about it is trying to do two things. Number one is to kind of systematically codify like what are the links between these different symptoms? For example, dopamine modulating therapies. Okay, you give somebody with Parkinson's a dopamine agonist or dopamine replacement therapy like levodopa. So what does that do? It helps you move better, causes all these side effects like hallucinations, increase in risk taking, hyperkinesia, even in Parkinson's patients. How do these different things relate to each other? Like, how is a hallucination related to movement initiation? It seems a little puzzling. And that was one of the things that I spent a lot of time thinking about when I was on the wards. And I think that maybe the difference in the way that we're trying to think about it is to try to combine the way that we thought about the movement system when I was a PhD student and looking at these dopamine ramps, which seem to be promoting this spontaneous generative process for movement, to try to draw analogies between that and these other types of processes, like a hallucination, for example, which could be thought of as a more extreme case of trying to make these noisy perceptual decisions, a more extreme case of imagining the lane lines in the road, as it were. And I think there's lots of evidence that dopamine is involved in these different types of processes. I think what we are aiming to do is to take what we observe in the motor system and draw an analogy with a neural circuit hypothesis that involves dopamine neurons and downstream neurons in the basal ganglia to try to understand how all these different types of more abstract cognitive and perceptual decisions might be analogized to these spontaneous motor decisions. So I'd say that that's kind of it. I wouldn't say that we're trying to knock over a prevailing view. I would say that we're just trying to think about it carefully and try to draw links, which I think is something that is missing.
Great. Thank you, Allison. Some more Parkinson's related questions are coming in. So one question is, what do we know about why the dopamine neurons die or disappear or have diminished activity in individuals with Parkinson's?
It's a really good question. I will tell you the things that I'm aware of, and I will also say this is a very intense field of research that has its own traditions and it has its own very deep literature, which I would have to advise you to go and read about that on your own. What I can tell you is that in Parkinson's disease, what we know is that those dopamine neurons die off. They degenerate for some reason. There are definitely genetic mutations that are associated with an increased risk of Parkinson's disease. There have been hypotheses that have been put out there that this has something to do with mitochondrial dysfunction, where these cells are just not able to handle oxidative stress, where they're not able to basically handle just typical stresses that happen to the neurons, and so they start to die out. But I think this is still definitely a very active area of research, what causes that degeneration.
And is anything known about why those neurons in particular are susceptible to that oxidative damage?
For that one, I don't have a great answer for that one. I'm sure that there are a lot of thoughts on that.
Yeah.
I have to look it up. I will look into it. Thank you.
Good. Back to some of the basic neuroscience. A question comes in. Do the decisions that these mice are making, do they require a consensus among multiple neurons? And if yes, how does the requirement for consensus affect your understanding of a probabilistic decision model?
Very interesting. Okay, I'm going to give you a couple of thoughts on that. So first, with the data that I've shown you so far, we don't have the ability from photometry signals to be able to look at individual neurons. So photometry is, without getting into too much detail, it's a population average, which means that the signals that I've shown you are basically the average light that's being emitted by a large population of dopamine neurons. We might be recording from dozens to hundreds, perhaps even thousands of neurons at a time, depending on where your field of view is and what your level of expression is. So we don't have an ability to look at that. Having said that, I think it's a very commonly said thing in the neuroscience field that, a single neuron doesn't make you move, right? Like this behavior is going to be the result and product of many, many, many neurons working together. Now, what can make this process probabilistic? I think based on what we can say, is there some sort of consensus between the population? Maybe so. I mean, it would be interesting to look at individual neurons. But what I can tell you about how we think that this probabilistic process arises is we think that basically dopamine neurons are feeding in a sort of heat underneath this generative process, which we think actually depends on downstream neurons in basal ganglia feedback loops that are feeding motor areas, higher motor centers. I didn't get into all the circuitry in this talk, but let me see, what can I tell you off the top of my head that would be useful to this? Basically, what we think is that potentially it's another area, like a higher motor center, that decides on what the movement is going to be and says, I think I want to make this movement soon, and starts sending activity through these feedback loops. What we think that dopamine does is it contributes to the process of when the movement initiates by putting heat underneath that feedback process to cause the system to rev up to the point of actually triggering the movement. We think that might be the ramp to threshold process that we're seeing happening here. And then what makes it probabilistic has something to do with the kind of the interaction between these two systems talking to each other, which sort of makes it seem from an outsider's view as being stochastic.
Mhm. Terrific. Sort of a question has come in that I'll relate to what you've just shared. You were referring to sort of downstream things that are downstream of the substantia nigra and the dopamine signal there. What is there? Are you giving consideration to the downstream circuitry, and do you have any insights into what that might look like? How do you get from those dopamine neurons and the substantia nigra? How do you get to movement?
Okay. So I have to show you the slide then. All right, so we're gonna do it. We're gonna get into the circuitry. Okay. Here we go. I thought about showing this today and I thought maybe it was too much. All right, so yeah, absolutely. In fact, our goal was always, and our goal in the lab right now is still to get downstream of dopamine neurons and to understand how the circuit works together. Because so far I've just shown you the activity of one set of neurons. In my mind, the exciting opportunity that we have with these new techniques that are coming out in the field of neuroscience is the opportunity to actually be able to listen to multiple nodes in these circuits to see how they work together. So indeed, so the initial model that I showed you guys can see this hopefully. Okay, so the initial model that I showed you is that, movements generate as a result of some burst of coordinated activity in these higher motor centers, which could be the cortex, it could be brainstem nuclei, what have you. It's that we're going to elide those details for the moment. The idea is that somehow something upstream of this has to cause these to become coordinated in the first place. So how does dopamine potentially fit into this? Well, we know that the dopamine neurons that are lost in Parkinson's disease project densely to an area of the brain called the striatum, which is one of kind of the main input layers of these basal ganglia feedback circuits. I probably can't get into all the complex circuitry today. I think it would be a little too much for this question, but let's see if I can kind of just draw this out for you, okay? So we know that dopamine neurons present positive input to direct pathway neurons that are part of this positive feedback circuit with the cortex and other motor centers. So basically, this pathway through a series of neural circuit steps causes increased activity to go to these higher motor centers. And then those higher motor centers then output more activity here. And so this is a positive feedback loop. And you can imagine like the ramping signals that we see in dopamine neurons, right? That kind of looks like a rev up process, which you could get with positive feedback. Dopamine neurons also simultaneously send suppressive input into this second feedback loop that is part of the striatum, which is the indirect pathway. And these neurons, unlike the direct pathway neurons, are part of this negative feedback loop. So, when you see positive and negative feedback put together, in electronics, that forms a control amplifier. So the idea that we've had for a long time is that what dopamine neurons could be doing is they could be causing the balance between these two feedback loops to tip in favor of the positive feedback loop. And you can imagine at baseline, right? You're not moving. Maybe the indirect pathway is winning, the negative pathway is winning. And so your higher motor centers are quiet. But as dopamine activity goes up, that would slowly maybe tip the balance in favor of the positive feedback pathway. That positive feedback could cause that revving up of these higher motor centers, causing this coordinated activity, this coordinated neural activity to seemingly arise out of nowhere, this spontaneous activity in the cortex. Now, our broader idea is that this kind of process doesn't have to be limited to motor types of spontaneous processes. This could apply to many different types of spontaneous processes because we know that this type of circuitry doesn't just interact with the motor system, it interacts with the perceptual systems, it interacts with more abstract cognitive systems in the prefrontal cortex and even emotional centers too. So we think this is kind of like the neural circuit mechanism that we're really trying to pursue. And we want to get downstream of dopamine neurons and start recording from multiple areas within these decision-making circuits to try to understand how they work.
Terrific, Allison. A question on that I think is intended to address your background in both neurobiology and your exposure to medicine through medical school. And it concerns the hottest drugs of the moment, the GLP-1 drugs. And the question is, do you see a connection between the phenomena that you're studying? Do you see a possible role for the new GLP-1 drugs, the hottest drugs of the moment, to help treat Parkinson's or other similar diseases? And what interactions, if any, might there be here?
What a great question. That's very timely. This is something that's definitely on our minds. So, these GLP-1 agonists, right? These were designed to treat type 2 diabetes, to help with, glucose homeostasis by increasing insulin secretion and, insulin sensitivity in the periphery. But amazingly, they're just these incredible weight loss drugs too. And the way that they, are thought to do that is by suppressing appetite. And one thing we know about GLP-1 is that one way it could suppress appetite is by slowing down gastric emptying. So you eat a meal and basically it would just move through the stomach more slowly and that would make you feel more full. So maybe that helps you reduce your appetite and eat less. But then there's all this other weird stuff happening with GLP-1 because it's not just appetite for food that's affected. It's also appetite for other stuff. So this is a lot of it's kind of anecdotal, but there's more and more evidence that this is a real thing, that patients on these drugs, instead of, not just decreasing their consumption of food, but they also decrease their consumption of addictive substances like alcohol, cocaine, amphetamines. Um, this has been studied in animal rodent models as well as, anecdotally in the clinic. And even more kind of abstract sort of things like some of the abstract decisions I was talking about, like gambling, these things are also seemingly affected by these GLP-1 agonists. Folks that used to do a lot of compulsive online shopping or going to the casino a lot, they feel like, well, I just don't really need to do that. So it's definitely on our minds that GLP-1 is interacting with these types of decisions, and that's another node in the circuit that we're planning on getting at. So, yeah.
All right, Allison, thank you for demonstrating the full breadth of your considerations and your thinking. And I'd like to thank all of you for joining us today. This concludes our presentation, and I'd like to end with a special thanks to Allison for sharing your work with us.
Thanks so much.
Thank you so much.